Nice to have you joining us once again on ECCMID Television. I'm Michael Jackson, your host, and with us in studio right now, a professor all the way from Switzerland. His name, Gilbert Greub, and he's the director of the University Hospital at the University of Lausanne in Switzerland. Gilbert, nice to have you with us, sir.
Nice to meet you.
Thank you. You've been an invited speaker at the conference. You've been talking on a couple of topics, on genomics and MALDI-TOF as well.
Yes, practically MALDI-TOF was a revolution in the diagnostic microbiology field because you can identify more than 95% of bugs just using these new tools. And this is a cheap, fast and accurate method. In addition, I was invited to speak about genomics just because we started a project where we want to use genomics in the clinical diagnostic. It's still not yet applied in most diagnostic setting. It's more research-based, but I think in the future it will become a great tool for clinical diagnostic microbiology as well.
I mean, it's quite exciting as well because both of these things are new areas, obviously, aren't they? We're learning every single day.
Yeah, we are on the learning phase.
Now, you were also involved in your professional career in discovery of new agents in pneumonia, and you also work in the field of miscarriage in the human form as well. Let's talk about pneumonia first. New agents in pneumonia, that sounds a bit scary.
Yeah, so you know about half of pneumonia are of unknown etiology. So the idea was to try to find out maybe new bugs that are not yet been discovered. So we are focusing especially on bacteria that do not grow without cells. And we are using different cell culture systems, including amoebal cells, tick cell lines and others, to try to find new bacterial species. And we find a few of them, including ones that we called Estrella lausanensis, for instance.
Many people in your field working in this particular area, sir?
No, there are not so many people, and there are lots of bacterial species yet to discover, and it's a fascinating area. And now we are mainly focusing on some chlamydia-related bacteria because we found several of these new bugs within the chlamydia-less order.
So you've been studying the biology of these chlamydia-like organisms, I understand, as well.
Yeah, we are studying both the biology and the pathogenic role. So regarding the pathogenic role, we discovered that Vaudois might be involved in miscarriage in humans. So, and regarding the biology, we are mainly interested in cell and molecular microbiology. For instance, how the bacteria is able to divide, what are the pathogenic or virulence factors that are expressed by these bacterial pathogens, what is the host-pathogen interactions, and this type of things.
So out of that original work, this area into the realm of miscarriage emerged for you as well. Is that how the two things tied together?
Yeah. It's just because we wanted to have some controls to know what is the basic seroprevalence in women that are healthy. So we took pregnant women, and to convince to do that, we also tested women with miscarriage and without miscarriage. We discovered that there was a striking association between antibodies directed against Vaudois and miscarriage. Therefore, we did an additional study looking at placenta, and in the placenta, we identified the bacteria by PCR and by immunohistochemistry. So this was quite impressive. And now the question is when to treat, how to treat this bacterial species. So we are looking also at that. And Vaudois is highly susceptible to macrolides, antibiotics, for instance, and it's relatively easy to treat with this type of drugs. So the question is now when to treat.
Yes.
Do we have to treat, for instance, after miscarriage to prevent a recurrent miscarriage? This is my proposal.
That's interesting. So all of your areas of work are quite groundbreaking, really.
Yeah, I think the chlamydia-related bacteria are yet unknown. There is really few people in the world working on that things, and I think there is lots of things to do. And for instance, for Vaudois, we use it not only because we think it is a pathogen, but we study it also because we use it as a model organism to study the biology of classical chlamydia. Chlamydia trachomatis, for instance, is associated with a huge morbidity. And the advantage with Vaudois, it's larger cells, so you can do subcellular localization of proteins thanks to these larger cells. And also, there are slight differences between Vaudois and Chlamydia trachomatis. So by studying these bacteria that grow much better and faster, it was possible to identify the protein involved in the division of Vaudois. And then we extended this work to the protein involved in the division of chlamydia. And all the division protein are putative targets for new antibiotics. And this is also a breakthrough that we published last year.
Awesome. And the response to your presentation, sir, from your colleagues here at ECCMID, how has that been?
It was a beautiful experience because it's always nice to be at the ECCMID. There is lots of interesting talk, lots of interesting poster presentation, and also we have a lot of interaction with the exhibitors, and because the exhibitors also do a lot of work in improving the technologies and the available tools for diagnostic purpose, for instance.
I've been asking a lot of the guests that have joined us here on ECCMID TV about how they work out their time at ECCMID. It's an incredible event, one of the world's biggest gatherings of your colleagues in the industry. How do you manage your time? I mean, I know you've been busy presenting, but do you spend time going to lectures, to oral presentations, to the plenary sessions, or is the networking more important to you?
I think it's difficult to manage your time because you have so many things to do. We have also some professional session. For instance, I'm in the subcommittee of the scientific affairs responsible for the diagnostic. So this morning I was from 7:45 to 11 blocked in a meeting.
Yes.
And also on Saturday morning I'm one of the associate editor of Clinical Microbiology and Infection, which is a journal of the society. And also all morning we were in another session, so discussing what will be the next special issue next year, how to deal with the peer review process and all these type of things. So yeah, we have a busy schedule, but it's a lot about networking, and we have also great time in the evening to discuss with friends, and it's I think we have just to try to split our time as best as possible.
I don't know how you fit it all in. Maybe we should make ECCMID 8 days in Istanbul. I mean, it's
yeah, but we have also to go back to work, and we have a
course.
lot of things to do there. So it's already a lot to be absent from our work for 2 full days.
I understand.
Of course, it's good that the meeting is on the weekend because it allows to have 4 days in total.
The learnings that you've picked up here, the contacts that you've made, all become invaluable to you in pushing the boundaries for the next 12 months, don't they, really?
Yeah, of course, it's not the only meeting we attend because there is also the specialty meeting.
Of course.
But of course, it's really important in order to push forward some things. You have new ideas, you have new contact, and we try to avoid to put too much in to have too much things to promote again because you are anyway busy when you come back. So,
of course. But certainly the catalyst for conversation and development thereafter as well.
It's a good motivation, and it's a catalyst, yeah.
Excellent. I understand that you've got an incredibly busy schedule. We're delighted that you took time out of it to come and join us here on ECCMID TV. Gilbert, it's been a real pleasure talking to you, sir. Enjoy the rest of your Congress.
Okay, thank you very much.
Thank you so much.
Thank you.
Thank you.